Hello mga katusok, kabunog, and kapuyat. Welcome to a day in my life as an intern. So as you can see, I am disinfecting my working area and also the donning of PPE. First, we need to make sure that all the disposable pipettes, all the test tubes are all labeled. As you can see, I am transferring the patient's plasma into another test tube to separate it from the red cells. Add as much volume of NSS until the mixture fills 2/3 of the tube. Mix it 3 to 5 times. Centrifuge the samples for 2 minutes at 3,400 RPM. Pipette and discard the supernatant on the sink. Make sure that packed red cells are not disturbed. After washing the red cells with normal saline solution, label 2 test tubes with 4 ml red cell suspension. Transfer 200 microliter of the washed red cells to tubes 1 and 2 with 3,800 microliter of normal saline solution. Thoroughly mix the red cells and saline. Place 1 drop of anti-A, 1 drop of anti-B, 1 drop of anti-D on a clean labeled glass slide. Add to each drop of reagent on the slide 1 drop of 2 to 5% red cell suspension of the red cells to be tested. Mix the reagents and red cells thoroughly by using a clean applicator stick for each reagent. Spread the mixture over an area. Gently tilt the slide from side to side continuously for up to 2 minutes. Place 1 drop of anti-A in a clean labeled test tube, 1 drop of anti-B in a separate clean labeled test tube, 1 drop of anti-D in a third labeled tube. And to each tube, add 1 drop of a 2 to 5% suspension of red cells to be tested. Then mix test tube. Add 2 drops of plasma to each clean labeled test tube. Add 1 drop of A reagent red cells to the tube labeled A, and add 1 drop of B reagent red cells to the tube labeled B. Mix the contents of the tubes. Centrifuge for 15 seconds at 3,400 RPM. Read macroscopically. Dilute 10 microliter of donor's red cell with 200 microliter of DG gel sol to prepare 1% of red cell suspension. Dispense 10 microliter of donor's red cell suspension to the corresponding well. And dispense 25 microliter of the patient's plasma into the same well. Incubate for 15 minutes at 37 degrees Celsius. Centrifuge for 9 minutes. Mix 2 drops of patient's plasma with 1 drop of 2 to 5% of donor's red cell suspension. Add 2 drops of LISS, mix, and incubate at 37 degrees Celsius for 15 minutes. Centrifuge for 15 seconds at 3,400 RPM and observe for hemolysis and agglutination. Wash the red cells 3 times with saline and completely decant the final wash. Add 2 drops of AHG to the dry red cell button and mix well. Then centrifuge for 15 seconds at 3,400 RPM. Dispense 1 drop of 2 to 5% suspension of red cells into a clean label test tube. Wash the tube 3 times with normal saline solution, then completely decant the final wash. Block dry using a paper towel to remove residual saline. Immediately add 1 drop of anti-human globulin to the cell suspension. Centrifuge for 30 seconds at 3,400 RPM. Examine the cells for agglutination, grade, and record the reaction. Add 2 drops of patient serum to a clean label test tube. Add 1 drop of 5% Group O cells red cell suspension in the same tube. Add 2 drops of LISS, mix and incubate for 10 to 15 minutes at 37 degrees Celsius. Centrifuge the tube for 30 seconds at 3,400 RPM. After centrifugation, resuspend the cell pellet by gently tipping the tube. Wash each tube 3 times with normal saline solution. Completely decant the final wash. Completely decant the final wash, then examine the cells for agglutination, grade, and record the reaction. This has been your intern, Queenie Thalia Maquidatu. Thank you.